Alzheimer's could be caused by brain cells that age too fast, according to a new study. Different types of brain cells could age at different speeds, and that may help explain the development of Alzheimer's disease. Engineers at the University of California, San Diego, UCDS, have used cutting-edge technology to analyze the post-mortem brains of 14 donors aged over 59, some of whom died with Alzheimer's disease and some of who did not. The team found that brain cells from the frontal cortex, where the most signs of aging and Alzheimer's disease shared a common feature. Their DNA deoxyribonucleic acid interacted with the genetic translator RNA ribonucleic acid in less intimate way than in healthy brains. The discovery was made using a tool called MUSIC, multicellular nucleic acid interacting mapping in single cells, which is powerful enough to peer inside a single cell's chromosomes to see how DNA and its associated RNA and proteins, collectively known as chromatin, interact. The technology has a potential to help us uncover novel molecular mechanisms underlying Alzheimer's pathology, which could pave the way for more targeted therapeutic interventions and improved patient outcomes, says bioengineer Shen Zhong from UCDS. Aging is known to cause changes in the chromatin of animal models, impacting an individual's lifespan, but the field of research concerning chromatin and aging is still new and rapidly expanding. Only recently were scientists able to create a 3D image of the true structure of human chromatin using super resolution microscopy. It looks quite different to how many textbooks imagined it. The new research from UCDS now sheds more light on the curious structure. Some brain cells in aging individuals look older than other brain cells. The individuals with more or other older cells were also more likely to have died with Alzheimer's disease. In cells with signs of degeneration, there were fewer short-range connections between chromatin and RNA. The erosion of these intimate interactions meant less contact for genetic translation, possibly inhibiting a cell's gene expression. This distinction sheds light on the correlation between the chromatin conformation of a cell and its transcriptonic age, thus extending our understanding from previous observations associating chromatin structural decline with aging to the single cell level. Researchers at UCDS conclude. While the study is small, it's interesting to note some sex differences that were found. Compared to the human male brain, the female brain showed fewer old neurons and more old supporting cells called oligodendrocytes. In further experiments among mice, researchers used UCDS found more oligodendrocytes showed age-related death in female mice than in males, indicating these brain cells were aging sooner. The same was not true of neurons. Healthy oligodendrocytes protect normal neuronal activities, and a balance between neurons and oligodendrocytes is required to maintain their bidirectional communications that ensure the necessary protectivity, explains researchers at UCDS. The team speculates that perhaps the faster aging of oligodendrocytes in female brains could account for why women are twice as likely to develop late-onset Alzheimer's. If we could identify the dysregulated genes in these aged cells and understand their functions in the local chromatin structure, we could also identify new potential therapeutic targets, hopes UCDS bioinformatics researcher Changzhao Wen. The study was published in Nature magazine. This is on Science Alert by Carla Casella. Please leave your comments and thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.